Perfect. Thank you, Mr. Koole. We're on. Thank you for joining me.
Yeah.
You are the project coordinator of the project LUTADOSE. Can you please introduce us your project?
Yeah, so thank you for the opportunity. So LUTADOSE is a project that focuses on improving the treatment of men with advanced prostate cancer. So basically patients at the end stage of the disease. And these are actually patients for who conventional treatments like chemotherapy are no longer effective. And to give you a bit of an idea about the clinical context, so each year up to 50,000 men in Europe could benefit from this type of treatment. It's called radioligand therapy or RLT. And this treatment actually uses radionuclides attached to a molecule that then binds to the prostate cancer cells. And this way we can deliver the radiation directly to the tumor cells while we try to spare most of the healthy tissue. And currently the two most types of radionuclides that are used are lutetium-177 and actinium-225. And so clinical studies have shown that this type of therapy works. For example, for patients treated with lutetium PSMA, so prostate cancer patients, the VISION trial has shown that these patients have a significant increase in progression-free survival. That basically means that these patients live significantly longer without the disease progressing. But there's still a major limitation, as today these therapies are given using a standard dose. So every patient is receiving the same activity, regardless of the differences in tumor loads or in organ sensitivity. And that basically means that many patients are likely undertreated because we are applying a very broad safety margin to avoid the toxicity in all patients. And so if you want to make this therapy more effective while still being safe, we need to make it a bit more personalized. And that is what we aim to do with LUTADOSE. We aim to develop new tools to monitor and measure how much radiation actually reaches the tumors and the organs at risk, so the sensitive organs, so that in the future, doctors can adjust the dose for each patient individually, rather than to use a one-fits-all approach. And to do this, we need new imaging and quantification methods to help us to estimate more accurately how much radiation is absorbed in the different parts of the body. And with these improved quantification tools, we actually aim to find the best possible balance between safety and efficacy for every patient. So that's a bit the introduction of the project.
Thank you very much. Second question, can you describe your consortium and explain how you selected and engaged your partners?
Yeah, so for the consortium, I will quickly share a slide. Let's see. So this is basically an overview of our consortium. We deliberately kept the consortium small, where we also engaged partners with complementary expertise and where also we had a proven history of working together. Many of us have collaborated together in previous EU-funded projects or through professional networks like the European Association of Nuclear Medicine or the European Federation of Medical Physicists. So there is a strong foundation of trust and efficiency within the consortium. And this allows us to move forward quickly also and to stay focused on the scientific and the clinical goals. When forming the consortium, we also specifically looked for partners who could cover the full translational chain, from clinical practice to basic science and technology development. And on the clinical sides, we have actually two partners. So that's LMU in Munich and the K Leuven in Belgium. So in Germany and in Belgium. And basically, these are two sites that are actively treating patients with this type of therapy, who also have access to the clinical imaging infrastructure, to patient data, and also to have some valuable medical input also into the project on how we can try to optimize these therapies like lutetium PSMA and actinium PSMA. And this also will ensure that the methods that we develop within the project, that these can also be easily translated into a clinical setting. So that's quite important that we have these clinical partners. But we also wanted to go beyond the clinic, and we also wanted to understand how the absorbed doses that we measure translate into biological effects, both in the tumor and in the organs at risk. And that's why we also included partners with expertise in radiobiology and computational modeling. So this is actually the SEK, so that's the Belgian Nuclear Research Center, but also the Erasmus Medical Center will give a lot of inputs on the radiobiology. And that's basically to better understand how alpha and beta emitting radionuclides, how they differ and how they damage the cells and tissues, and how, based on this information, we can give a better interpretation on the quantitative imaging data in terms of clinically meaningful dose estimates, so that we can basically better assess the risks to the kidneys and to the salivary glands, but also better assess the therapeutic effects to small and heterogeneous tumors. So it's actually this combination, this consortium, that will allow us to combine different types of expertise and to take an integrated approach, so that we are not only improving what we see and what we measure, but that we also try to understand it and to make these treatments more effective and safe.
Oh, thank you. What key results do you expect from the project, and how do you foresee these outcomes impacting the EU community?
Yeah, so we are expected to have a number of concrete and clinical relevant outcomes that will help us to advance the personalization of radioligand therapy across Europe. Our main goal is actually to develop and validate quantitative imaging protocols for both lutetium and actinium, where we will also include the next generation of 3D CZT SPECT-CT system. So that's a new type of imaging system that can be used for measuring the activities within the patients while they are undergoing the treatment. And these protocols will support more accurate and reproducible activity quantification, which is essential for moving forward from this fixed dosing to really individualized therapies. Alongside that, we will also not only focus on the imaging, but we will also standardize the dosimetry workflows to actually go from the imaging data also to estimating the absorbed dose to the tumors and the organs at risk on a patient-by-patient basis. This also includes advanced segmentation methods to segment the organs, the tumoral lesions, also to validate the kinetic modeling approaches. And since it's very challenging to image actinium-225, we also want to translate some of the information that we get from the lutetium to the actinium, and to basically consider lutetium as a surrogate for actinium in a clinical setting. And that is on the patient level, but we also want to really focus on the biological level. And as mentioned before, also to better understand the biological effectiveness of these treatments. And there we will improve our models to basically better compare alpha and beta radiation effects, where we will also include like the microscale energy deposition and also the recall of radioactive daughters, especially with actinium-225. And this will allow us to refine dose effect relationships so that we have a better understanding of which dose has which biological effect, and ultimately to translate the imaging and the dosimetry data into clinically meaningful predictions of response and toxicity. So that's basically what we expect as impact for the patients in Europe.
Thank you. Last question. As your project has been running for about a year, what would you highlight as the most significant achievement so far?
Yeah, so since we are running now for 1 year in the project, this is basically the 3 work packages that we define for the project. And for the first year, we basically focused on work package 1, which is focusing on the imaging part. And there we actually did a lutetium intercomparison study between different SPECT-CT systems and different clinical sites. I also can share you a slide here. So we basically we made different reference files and phantoms we assembled these phantoms and vials in Leuven, and then we actually shipped them to different sites in Belgium, so that we could cover different types of SPECT-CT systems, but also different activity meters. And also the activity meters are quite important because these are the measurements that you will use to actually quantify the activity that is administered to the patients. So this is actually what we have done in the first year. And then we used these files to actually check the activities that were measured at different clinical sites, and we compare them with the reference values that we had. And also for the SPECT systems, we also had this uniform phantom where we could check actually the calibration of the SPECT system. And we also had this image quality phantom, which is basically a phantom that consists of hot spheres on a cold background. And based on this phantom, we could actually check the image quality. So basically, we have collected all these data, and we are now in the process of analyzing them. And so that will also be something to make sure that the imaging performance is on the same level across different clinical sites in Belgium. And one thing that we already demonstrated with this study is that if you use a new generation of SPECT-CT systems, like the CZT systems, we could actually demonstrate that you could reduce the scanning time for patients up to a factor of 3 compared to a conventional system. So this has also, again, strong implications for patient comfort, but also for the clinical workflow. In addition, we also developed some segmentation tools for the organs at risks and for the tumoral lesions. And these segmentation tools will also not only be used for estimating the dose to the organs and to the tumors, but we will also use these segmentations to use the SPECT data, so the lutetium SPECT data, to monitor the therapy response. Because if we can reliably segment all the tumoral lesions, we have an idea of the total tumor volume. And based on the reduction or the increase in tumor volume during the treatment, we can monitor how the patient is responding to the treatments. And that is in addition to the SPECT scans that are sometimes performed as well, but not on a regular basis. And then going back to the overview for work package 2, we are also now initiating some studies. This is basically more focusing on the radiobiological components. We have initiated the labeling procedure with actinium-225. We have also initiated the cell lines. We are now starting with some initial cell experiments. And in parallel, we also now refined our cellular dosimetry models. In the past, we used a very straightforward spherical model where we approximated the nucleus as a sphere and also the cell as a sphere. But now we have more realistic cell geometries based on microscopic data. So we are hoping there that we can more realistically simulate the dose to the cell nucleus. And finally, in work package 3, we are also now working on geometrical models of the organ microstructure to better understand how the short range of alpha particles will affect the functionality of the organs at risk, but also how effective these alpha particles are with killing the tumor cells because of the very short range. So altogether in this first year, we have built a strong foundation for the next 3 years. We have been validating the imaging performance. We have been validating the segmentation and the dosimetry tools. We have also initiated the radiobiological studies. And the next steps will be to further build on these tools and do some further developments. So that's basically what we achieved in the first year.
Quite a lot, I would say. Thank you very much.
Yeah. Okay.
Is there anything else you would like to add?
No, we just want to thank Pianoforte for the opportunity to do this research because we think it's quite important for the patients and for the healthcare in general in Europe.
Definitely, it is. Okay, Mr. Koole, thank you very much for your presentation and for your time. It was incredible. Thank you.
You're welcome.